Cyclin B gene, or CCNB1, is a gene found in chromosome 5. Its exact location is 5q13.2, the Q meaning that it's located in the longer arm of the chromosome. This gene encodes for the whole protein G2 mitotic specific cyclin B1, also known as cyclin B1, but there are other genes which encode for other types of cyclin proteins. But then, what are cyclins? In order to understand them, we need to know what the cell cycle is. It is a process of 4 phases that cells pass through to allow them to divide and produce new cells. These phases are G1, in which the cell grows physically larger and copies organelles, S, or synthesis phase, where DNA replicates, G2, in which all the enzymes needed in the process of cell division are produced, and the last phase, M, or mitosis, in which the cell divides its copied DNA and cytoplasm to make 2 new cells. Now, what are cyclins? Cyclins are cell cycle regulators. Each cyclin is associated with a particular phase or set of phases in the cell cycle and helps drive the events of that phase. As you can see in this chart, the levels of the different cyclins vary considerably across the cell cycle. A typical cyclin is present at low levels for most of the cycle, but increases strongly at the stage where it's needed. Cyclin B, for example, peaks dramatically at the transition from G2 to M phase. These cyclins are unusual in that they are needed for much of the cell cycle. In order to drive the events of each phase, a cyclin must activate or inactivate many target proteins inside of the cell. They do that by partnering with a family of enzymes called the cyclin-dependent kinases, CDKs. Alone CDK is inactive, but the binding of a cyclin activates it, making it a functional enzyme and allowing it to modify target proteins. And how does this work? Well, CDKs are kinases, enzymes that phosphorylate, attach phosphate groups to, specific target proteins. The attached phosphate group acts like a switch, making the target protein more or less active. What is their structure? All members of the cyclin family are similar in around 100 amino acids that make up the cyclin box, which are organized into 5 helical regions, meaning that it has the form of a spiral, and are important in binding partner proteins, including the CDKs. And specifically, how does cyclin B work? Cyclin B is necessary for the progression of the cells into and out of M phase. It stays at low levels for much of the cycle but builds up as the cell approaches the G2 to M transition. As cyclin B accumulates, it binds to CDKs already present in the cell, forming complexes called MPF, maturation promoting factor, that are ready to trigger M phase. Once these complexes receive an additional signal, essentially an all clear confirming that the cell's DNA is intact, they become active and set the events of M phase in motion. The MPF complexes add phosphate tags to several different proteins in the nuclear envelope, a part of the nucleus, resulting in its breakdown, and also activate target proteins that promote chromosome condensation and other M phase events. In addition to driving the events of M phase, MPF also triggers its own destruction by activating the anaphase promoting complex cyclosome, APCC, a protein complex that causes cyclin B to be destroyed starting in anaphase, the third stage of mitosis. Now, which cells express the CCNB1 gene? Every cell contains the whole DNA of the organism, but only some of them express this specific gene. Every cell that undergoes cell division needs to express the CCNB1 or cyclin B gene. Cells that don't divide, such as neurons and muscle cells, won't express it. Cyclin B's role in cancer. Cyclin B plays an integral role in many types of cancer. Hyperplasia and controlled cell growth is one of the causes of cancer. Because cyclin B is necessary for cell division, cyclin B levels are often deregulated in tumors. When cyclin B levels are elevated, cells can enter M phase prematurely and strict control over cell division is lost, which is a favorable condition for cancer development. On the other hand, if cyclin B levels are depleted, cells cannot enter M phase and cell division slows down. Some anti-cancer therapies have been designed to prevent the formation of cyclin B CDK complex in cancer cells, to slow or prevent cell division. Thanks for watching.